Hello, my name is Dr. Eric Landrum from the Department of Psychology at Boise State University, and this is my research method screencast about data collection issues. I think this is actually, although on the surface it might seem pretty dry, this is one of these topics that I think that all psychology majors, whether you're going to graduate school, you want a good job with your bachelor's degree, you could really apply this in your future as you're thinking about helping organizations, collecting data, interpreting data, and really being a critical thinker. So I think these topics have some really broad practical applications. So just to start big and broad about with an overview to talk about artifact. Let's just say from the outset that an artifact is not a good thing. It's not a desirable outcome. Our goal is to design studies in which the dependent variable is a nice, clean, meaningful measurement of the construct we're interested in and not an artifact. So if we have an artifact creep in, that means there's some sort of distortion or bias of the measure. So this is a classic study from decades ago, and this is actually real work based on real research. So college students were trained in an experiment on the effects of food reinforcement on the rats' speed and accuracy in learning a preference for the dark side of a T maze. And so I'm going to show you some graphics about this in a minute, but it kind of looks like this. So you see the start there, and somebody thankfully put up a little graphic on the internet, which I grabbed for this. And so the rat starts there, and the goal is to get them to turn left or turn right, but to go from a lighted area into a darkened corner, either the left direction or the right direction. And rats, generally speaking, in this scenario, do not desire to do that. So I just want to flip back here. So the idea is that can you use food reinforcement on a rat's speed and accuracy in learning that preference for the dark side of the T maze when that's not their natural preference, all right? So that's the goal. Get them to turn right or turn left, whichever one is dark. You can use food as a reinforcement at the end of the little runway there. But here's what the experiment was really about, which is really interesting. So some of the participants in this study, so remember, each student is kind of like the team leader and has a rat that's going to be placed in this team maze. And so half of the students were told that their rats are, and you can see it in quotes and in boldface here, maze bright. All right, and so they were told they were maze bright. They should do really well on this task. After the study was over, when those students were interviewed, they said that their rats tended to be more bright, pleasant, and likable. I'm not making this up. All right, so that's what they were told. In another condition, other students were told that their rats were genetically inferior or maze dull. And so after the study was over, you asked those students to describe their rats, and they used terms like dull or unlikable. And so hopefully you can kind of see ahead to how the study turned out. The rats were genetically as identical as they could be, from the same litters, from the same strain, species. And so they were as close as they could be within the rat world. The instructions were the independent variable, where half the students were told one thing, half the students were told the other thing. So as you can see, even already, when you ask students about their descriptions, they bought into it because the first group said that they were bright, pleasant, and likable, and the second group said they were dull and unlikable. Now, interestingly, it's the experimenter, their perception and their treatment, whether purposeful or accidental, their treatment influenced the level of enthusiasm with the rats, as well as the rats' speed and accuracy in learning the task. So the rats that were in the maze bright condition, not only did they make the turn faster in the T maze, but they made fewer errors. You can imagine you go down the wrong way, that's one error, then you go back down to the stem, that's two errors. There's multiple ways to make an error, even in a pretty simple T maze. Experiment was really about what the students were told about their rats. And so that's actually kind of interesting. So just that framework, just that framing, those college students influenced either positively or negatively the speed and accuracy of the rats running in the task. And I think that's really an important takeaway from this entire screencast about data collection issues. The way we treat our participants might influence their potential outcomes, the measures that we collect on the dependent variable. Now, we've talked a little bit about this before. We talked about survey research. We talked about the way that you ask the questions can influence the answers. But this is even, I guess, more broad than that. Even the way that you treat or address or your attitudes or opinions about your participants might actually influence your participants' behavior. And by golly, if it happened in rats, for goodness sake, you can imagine the influence that one human can have over another human. So, what psychological researchers want to do is we want to minimize these artifacts. I don't want you behaving in my study the way you think I want. I want you to behave naturally and the way you would if you weren't in a study at all. So we actually want to avoid these artifacts. And unfortunately, we are spending an entire semester thinking about research methods and the ways that we can do that. And this screencast will give you some very specific examples. So one type of artifact, one kind of class or category of artifacts are called physical setting artifacts. These can affect tension, comfort, and concentration. So if you are in a study where you're in a college classroom, a lecture hall, and you're filling out a survey, or you're asked to show up at some location on campus, or maybe even you're doing an online survey in your bedroom at home, the physical setting can certainly influence your performance on certain tasks. And that might be noise level, time of day, even the pre-research atmosphere if you're in a face-to-face study. What the experimenter is talking about at the front of the room. Are they being cordial? Is it kind of a cold, clammy feeling kind of thing? Even the temperature in the room, degree of crowdedness, the density. You've probably taken tests maybe back in high school where everyone was jammed into a room versus in a college lecture hall. You might like to spread out and have every other space be an empty space in the classroom. And so the physical setting can actually influence performance on dependent variables. And when that happens, that's a real threat to external validity. Now, we talked about types of validity in a screencast a while back, if you're watching these in order, of course. But the notion is that external validity is this ability to generalize from my study to other people, populations, times. And so if the physical setting is making a difference, that will limit the extent to which I can generalize that study beyond my sample that I studied. So there are a couple different techniques on how to avoid this artifact. And so one would be constancy, another would be blocking. And so, for instance, if I'm worried about time of day, let's say, so if I want to conduct a study and participants have to come to a classroom on campus, and I'm worried about time of day as a physical artifact, I won't do a session at 8 a.m., 12 noon, and 4 p.m. I'll do a one-shot study where they're all done at the same time. That would be an example of constancy. Or blocking. And so if I really think time of day might be an influential variable, I might go ahead with my 8:15, 12 noon, and 4:30 study, but I'm going to count time of day as a variable, and then I can actually try to test and see if it had a significant effect on the outcomes of the study. I think part of this is going to be you need to address, do you test participants in groups or as individuals? With individuals, there's lots more variability, although it's more convenient for the people. It's pretty inconvenient to have, okay, I need 100 people to show up at 4:15 on Thursday afternoon to be part of my study. So again, it's all about trade-offs between the ease for the researcher and the convenience for the participants, because if it's too complex for the participants, they're not going to want to show up. Even if you were to pay them, they might still not want to show up if it's just going to be too inconvenient. So that was one class of artifacts called physical setting. Another one's called within subjects. You see here at the top of this screen. So what response set acquiescence is all about is the pattern of responding to usually survey questions would be kind of the typical scenario. So response set is a certain pattern, but the acquiescence is that the pattern follows on one end of the spectrum or the other end. In other words, if you have a bunch of questions and I start answering strongly agree, and the next one is strongly agree, and I answer the next one strongly agree, the notion here is that the fear is that the participant stops thinking about the individual questions but starts paying more attention to the pattern. So rather than reading each question carefully, they already know they're going to strongly agree because they've already strongly agreed for the first 4 items. And we clearly don't want this. We want them to read every question and answer it as honestly and truthfully as they can. We don't want this shortcut to occur. That's why it's an artifact. And so one way to do that is to be careful with the wording. And so, you can phrase things positively, but with the answer being no or strongly disagree. I think I've got some examples coming up here for you. So you want to mix up the pattern of responses in order to encourage each participant to read each item very clearly. Good. So here are the examples that I thought were in the screencast. So if I'm doing an end of semester a teaching and learning evaluation on my course, and I asked these 4 questions, the instructor held my attention, wrote exams that were fair, the instructor seemed well prepared for class, was available for extra help. So if you're going strongly agree, strongly agree, strongly agree, strongly agree, the notion is that you might stop reading the next items after this and then just kind of go on autopilot and start bubbling in or selecting on the radio button strongly agree. So to avoid that, it would be much better to ask a sequence like this. And so the instructor was seldom able. So a positive answer to that would be to disagree. The instructor wrote exams that fairly tested material. Again, a positive answer there would be to agree. So now you can see a great pattern. If to be a good instructor, the desirable pattern would be a zigzag. And so I want you to strongly disagree to this one, but I want you to agree to that next one. And so you can kind of shake things up by making sure that not all the positives are on one side of the scale and not all the negative responses on the other side of the scale. You can see the example here with the wording I provided. This is also another version or flavor of within subjects artifacts, response set social desirability. And that means that participants are not accurately reflecting themselves and their opinions, but they present themselves as socially acceptable. And this, especially with research topics that are a bit sensitive or might be a hot topic, this actually happens fairly often, and we want to be careful with this. So, for example, if I was doing a research study on racism, I'm looking for sources of racism and influencers and, the role of parents and media, society, schools and everything, professors and all that. I'm probably not going to do a study in a classroom where I say, okay, raise your hand if you're a racist. That's not really a socially desirable answer, even if really in our heart of hearts, I kind of tend to believe, and I've said this in a prior screencast, that we are all a little bit racist at some point, at some level, some more than others, but we all have that knee-jerk reaction sometime to someone who from a different culture than us, or looks different from us, or speaks differently than we do. And so it would be really hard for someone to come out and say, yeah, I'm a racist, or think about other really sensitive topics, sexual predators, child molesters, people who perpetrate and commit domestic violence. I mean, they're not going to raise their hands. These are really important topics, and it's important for us to study, but we're going to have to be subtle and nuanced about it. People, generally speaking, on a survey, even if they believe it's anonymous, are probably not going to be anxious to admit to illegal behaviors. And so these topics are really important, but we have to be savvy as researchers so that we can try to get at them as best that we can, because we don't want this to happen. We don't want people to do impression management and show us how they think they should look. We want them to show us how they really are. So how could you tell? Well, there are some research approaches to this. Here's just a really quick example. If I were to ask you these 3 questions on a survey, I sometimes feel resentful when I don't get my way. I am always careful about my manner of dress. I like to gossip at times. The most honest, to be clear, the most honest answers, the pattern would be true, false, true. Come on, don't we all sometimes feel resentful? I am not always careful about my manner of dress. And yeah, I do like to gossip at times. So it's not just one item, but it's a pattern of items. It's a pattern of multiple items. But the social desirability pattern would be false, true, false. So if you were to ask your survey questions and you tacked on these last 3 and you saw false, true, false, that would be a red flag. That would be an alert that says, I need to, think about, do I want to keep this person in the study? This might be an outlier. I'm not so sure they were giving me honest answers on the other part, because it seems pretty clear they were giving me social desirability answers on this part. And by the way, there are entire scales. There's a classic Marlowe Crown social desirability scale. there are social desirability scales built into major products like the MMPI-2 and things like that, where you can actually get a measure of one's level of social desirable responding. So the approaches, and I think we've kind of inferred these. So one thing you want to make sure that you want to convince folks that that their participation is anonymous. And so if you're asking about something sensitive like racism or domestic violence, if you want to get at those honest responses, one way to encourage that is to really assure and go to great lengths to tell folks that their data are going to be anonymous after the study is over. And if you can, and this is tricky sometimes, you make socially desirable options, I'm sorry, make socially undesirable options as acceptable as socially desirable options. So I might not come out and ask the question, are you a racist? But I might ask a series of questions that kind of hover around, do you always treat everyone the same? Do you treat people from some places differently than you treat people from other places? And so you're not really coming out and using the words per se, but you're trying to kind of tiptoe around what might be an emotionally charged word like racism or domestic violence or something like that. Again, important and topics to study, but a little bit tricky because it's hard for people sometimes to be honest about those really challenging behaviors and situations and occurrences that do happen. The MMPI-2 uses what's called the lie subscale. And so if I were to ask you questions like, I always think of other people first, I have never had an erotic fantasy, and there's a whole bunch more. If people are always saying yes to these types of items, and think about it, it's the always and the never. It's really not about the other people or fantasy. I always think of other people first. I have never had an erotic fantasy. So if you catch people in a pattern, their answers may have less validity. You may have to take those with a grain of salt, so to speak. And like I already mentioned, the MMPI-2 uses a lie subscale. All right, I want to tell you a classic story about within subjects, and your textbook goes into this a little bit more, and I want to just kind of cover the highlights of it. But two researchers from the University of Minnesota in the late 1920s into the early 1930s did some studies at a Western Electric Hawthorne plant in northern Illinois, and they were interested in improving productivity. And the graphic you see here on the screen on the right frame is like a telephone relay assembly from the old times. So there's this big plastic metal phone, and there was the rotary dial, and there were components that were mechanical in it, and so there were these electrical switches that had to be assembled by hand. And in this particular set of studies, at least for the one I'm going to talk about right now, you're talking about 6 female assembly line workers. And so the notion was, how can we increase productivity? How can we get more units per hour? How can we get less fatigue, taking fewer breaks, having fewer units with mistakes and errors, that kind of thing? And so these two researchers went to the Hawthorne plant, and they pulled these women out of the normal assembly line and put them in a special room so they could measure and watch and observe and count the number of relay switches that were made per person. All right, so that's actually the dependent variable, the number of telephone relay switches produced. And of course, you can see offshoots on that. I could look at productivity, the number per hour, accuracy, the number of correctly operating switches per hour, and so on. And again, this was a series of studies. And here's kind of a glimpse, a picture from the time of, yeah, all 6 women are there. And you can see they're set in a room, and they've got all the stuff in front of them, and, the products, get swept into a bin in front of them. And so you can kind of see how that dependent variable is measured. Now, the independent variables, and there were, again, you're talking about a series of studies over 6 or 7 years, a bunch of different things were tried. And so some independent variables were designed to increase productivity, and some independent variables were designed to decrease productivity. And that actually kind of makes sense if you think about it. If I know what decreases productivity, I want to make sure I don't do that in my assembly plant. Okay, if I do know what increases productivity, and if it makes sense, then maybe I do want to try to do those things. So we want variables that go in both directions, so to speak. So what helps productivity, what hinders productivity. And so you can see here on this screen some of the variables that were tried to decrease productivity. These were usually done for at least 2 weeks, and so you might start 2 weeks in, and then for week 3 and 4, we're going to dim the lights, and we're going to do that for 2 weeks. Then the next 2 weeks, we're going to shorten the workday by 1 hour. All right. Then the next 2 weeks, we're going to increase the number of coffee breaks. And again, remember, we got this dependent variable of counting the number of telephone relay switches. And so you can see the different variations. And sometimes there are other parts of the study. It's very complex. They might let them reset to normal, and then the next intervention would be increasing the length of the workday without rest periods or coffee breaks. So you can see the researchers were interested because they believed that all of these interventions, if you will, conditions of the independent variable, would decrease the productivity of the 6 assembly line workers. There were also, as you see at the bottom of the left frame there, independent variables designed to increase productivity. So for a couple of weeks, they got a pay increase. They got a raise in their wages. Another 2 week block or longer, they had optimum temperature. So, the right amount of heat in the winter, the right amount of cooling in the summer. In another block of the study, they got optimum illumination. All right. And so this is almost like a push pull. The idea being, can we manipulate the output and the productivity by manipulating the variables? These variables, physical, environmental variables, or coffee breaks, rest breaks, bathroom breaks, into the nature of assembly line work. All right. So interestingly, and it's kind of fascinating, and this is actually called the Hawthorne effect, the hypotheses were not supported. Remember, we had some hypotheses that predicted an increase in productivity and some that were designed to predict a decrease in productivity, but that didn't happen. And this is the really cool part of the study. No matter what these researchers threw at these 6 assembly line workers with regards to independent variable manipulations, they always increased. The workers always stepped up to the plate. You gave them optimum illumination, they did great. You made them work an extra. You took away an hour of the workday. Not only did they work great, but they got better. Over time, they got better and better and better, no matter if you gave them a variable manipulation to increase or decrease their productivity. And the notion was these 6 assembly line workers, they felt honored. Out of the entire plant, they were selected to represent, their fellow employees. The employers had taken special notice. There were these 2 researchers from the University of Minnesota hovering around all day long, watching what they were doing. It's like this team spirit evolved. We don't want to let them down. And so the variables that were designed to decrease productivity actually backfired. This feeling of specialness by being part of a study in this new situation, in this secluded environment, came to be known as the Hawthorne effect. And so, again, this is an artifact. We do not want our participants in a study to band together and let's try to do our best, unless that's what the study is about. And so we don't want people to change their own behavior as a function of being in the study. And that's what I mean by within subjects artifact. We don't want them to change from within on their own. To be honest with you, as a researcher, I want behavior change because of my externally delivered independent variable, not that the people changed on their own over time. Another artifact in our hit parade of artifacts here is called demand characteristics. And this is kind of interesting because, again, it's another change in the participant, but it's not just their own within subjects self-motivated change. And so, and sometimes this term kind of confuses folks. So the demand characteristics, this notion is from the perspective of the participant. And essentially what it means is that sometimes participants are eager to please and they try to figure out the demands of the study, or said another way, they try to figure out what the experiment wants or is trying to find. And sometimes the participants will act in a way to be pleasing to the experimenter as opposed to acting the way that they would normally act. So, the way to think about it, sometimes the participant is eager to please, and sometimes the researcher is eager to be pleased. And so, and again, we don't want that. We don't want them to change their behavior because of their own ideas, or we don't want them to change their behavior because that's what they think the experimenter wants. So if participants detect they're in a special group, their interest may increase. We call that the Hawthorne effect. If participants detect they're in a special group, their interest may increase, and they're going to save the day no matter what. We call that the Hawthorne effect. But if those participants detect they're in a control group, their interest may wane. You see this sometimes in weight loss studies. And so if you're in the experimental group and you're losing weight and the product is working, that's all well and good. But if you're in a weight loss study and it's not you're in the control group and you're not losing weight, then you might lose interest in the group and you may in the study and you may drop out. And again, here's a great little reminder from the Mark Wahlberg research meme about demand characteristics. We certainly don't want this experimenter bias. So one approach to help avoid demand characteristics is what we would call a single blind study. So in a single blind study, the participants are uninformed as to whether or not they're in the experimental group or the control group. Now, you've probably heard double blind before, and we'll get to that by the end of this screencast. So in a single blind study, I'm going to know as the researcher who's in the treatment group or who's in the control group, but the participants themselves won't know. At least they won't be told. Sometimes they figure it out, but they won't be told by the researcher. Sometimes if you want to try to figure out, did my behavior as the researcher influence your behavior as a participant? Another way to see if demand characteristics were operating would be to do a post-experiment questionnaire or interview and ask the participants, did you change your behavior at all because of anything I said or did during the study if I'm the researcher? And so that's just kind of another way to help confirm whether or not demand characteristics were in play. And again, it's an artifact, so we don't want folks acting the way we want them to act. We want them to act normal and natural, and we just want to measure that behavior as it happens. Another very related artifact is called experimenter expectancy. And that's kind of related to demand characteristics. It's a bias as a function of the researcher's motivation. So, if I'm doing this study, and we've talked about this in other screencasts, if I'm doing a study, I have a bias to reject the null hypothesis, and I want my study to work. It's hard for me to publish and advance my career with a bunch of results that say, I found nothing. And so there is expectation on the behalf of the experimenter to look for differences in the study or significant correlations or whatever you're looking for. And so I am expecting a difference. And so hopefully I'm not treating people differently. So demand characteristics is really about, is the participant trying to figure out what the researcher wants? Experimenter expectancy is about, is the researcher accidentally or even purposely giving hints, tips, or clues about what the researcher wants and then acting accordingly. And I've got there's a great story about Clever Hans. I'm not going to tell that story here. There's a book that was recently written about it, just a really nice book. There's a screenshot up there. The long and the short of it was a farmer in Germany in the late 1800s, early 1900s, thought that his horse had higher intelligence than normal, could add, subtract, understood language. And in fact, what was happening is that subtle cues and clues were being given to the horse to behave in a certain way that looked like intelligence, but it really wasn't. And so it's a great story. Google Clever Hans or pick up that book. So, and you've heard me say this phrase already in this screencast, often research participants are eager to please and researchers are eager to be pleased. And so some possible examples of experimenter bias, if I'm overly friendly before a study happens, if you're in the room and, if you're answering questions from me and all of a sudden I tense up when I hear an answer I don't want, are you going to pick up on that cue and change your answer or be more nervous for the following questions? Do I walk over to the left side of the room and treat them one way, then walk over to the right side of the room and treat them another way? Is that an indicator of experimental group, control group? Am I really emphasizing certain words when I'm giving the instructions that may be an unintentional tip or hint or clue as to how I want you to behave? And then even the word choice in instructions or questions. In fact, when we looked together at the survey design and the survey screencast, we know that the way the question is asked can influence the type of answers that you get. So we know that word choice is critical here. So there are a couple of different approaches to minimizing experiment or expectancy. Probably the classic one that you've heard of is the double-blind study. And so this is interesting because in this study, the participants don't know what group they're in, but neither does the researcher interacting with them. So if you're in that classroom and one half of the participants in the classroom are getting the treatment, and the other half are getting the control, and I'm the experimenter in the classroom, I'm just running the study, but I don't know if you're what condition you're in, so that when you ask me, I can't lie about it. I can't give you hints or tips or clues because I don't know. Now, somebody else who I'm working for in this research lab setting knows who's getting what, who's in what condition. But if I'm the one interacting with participants in this way, if it's double blind, they don't know what condition they're in, and I don't know what condition they're in. Therefore, I cannot give them these experiment or expectancy tips or hints, even if I'm trying to avoid doing that. If I don't know, I don't know. You can train researchers to ensure standardized treatment of participants. Sometimes we call this a one-shot study. If you are worried that the instructions were going to be different for different groups of participants, you could tape record and play it, or you could do it online and make every participant read the exact same text before they launch into a study. So you can do that one-shot session, make sure everyone's treated exactly the same. And that's it. And I think those are some pretty interesting data collection issues. And, we're going to use our research methods to help avoid any pitfalls we might fall into when interpreting our data. And that's it for me and this research methods screencast.